Hello, everybody. We all are going through a very bad phase, and I hope you all are doing good. I am Rida, and today I'm here to present my presentation. And the topic of my presentation is factors affecting column efficiency. Now, the first question that arises in our mind is what is chromatography and precisely a column chromatography? So, let me give you a brief idea about a chromatography, column, and a column chromatography. So, chromatography is an important biophysical technique that enables separation, identification, and purification of the components of a mixture for qualitative as well as quantitative analysis. It is a separation technique in which a mobile phase carrying a mixture is caused to move in contact with a selectively absorbent stationary phase. Moving on to a column. Column is a tube-like structure. As you can see in the picture, it is a tube-like structure that is either made up of glass or plastic. The main components of any chromatography are a stationary phase, a mobile phase, sample, and the column. And in case of column chromatography, stationary phase is a solid and mobile phase is a liquid. In column chromatography, purification is carried out with the help of a column. That is why it is called a column chromatography. Let's move on to our next slide and our next topic, that is column efficiency. Column efficiency, also known as a plate count, is a measure of dispersion of a peak. It is expressed by the number of theoretical plates and is determined by the given formula. And in this formula, TR stands for the retention time measured from the instant of injection, and W stands for the peak width. It is the peak width. Fine. The number of theoretical plates is a measure of goodness of the column. That means more the number of theoretical plates, better is the column efficiency of the column. Now, another important point that we need to remember is that if the retention time is high and peak width is narrow, then it shows excellent chromatograms. Let's move on to our main topic, that is factors affecting column efficiency. This was all about the introduction to chromatography and column chromatography. Now is the main topic of my presentation, that is factors affecting column efficiency. There are several factors that affect the efficiency of the column, such as dimension of the column, its temperature, flow rate, packing, particle size, absorbent, etc. I'll discuss each of them separately and give you a brief idea about each of them. Starting with the first one, dimension of the column. Columns of various dimensions are available for use. Depending upon the process suitable, column dimensions need to be selected. Fine. Height, length, and the width ratio is an important factor. Increasing the length of the column, it has shown to have improved the separation or the resolution. Fine. Mostly for desalting purposes, long columns have shown to give satisfactory results, but optimization is needed for each one of them. That means we have to make the best use of the resource to get effective results. Fine. The examples of certain columns are XK columns, jacketed column. So let me just tell you what a jacketed column is. Jacketed column is one which has indentations on the inner tube to support packaging material. Fine. So the examples are XK 16/40, XK 50/30. Here, let me show you. Here, 50 stands for denotes the inner diameter of the column. That means this, and it is given in millimeter. Fine. And 30 is the length of the column, that is given in centimeter. Fine. 50 is the inner diameter of the column given in millimeter, and 30 is the length of the length or height of the column, and this is given in centimeters. Fine. I hope this was clear. Moving on to our next factor, that is the particle size of the absorbent. If the size of the particle decreases, separation is improved. That means the particle size and the separation, they are inversely proportional. Fine. Third important factor is solvent. Solvents used in column chromatography, they should not affect the stability of the proteins, or it should not cause any deleterious effects. Solvents to be used should be compatible with the column and the matrix. That means solvent should be compatible with the absorbent as well as with the column. Usually a solvent in column chromatography is used which is less viscous, with less viscosity, as the viscosity can influence the flow rate, the solvent system, and which affects the separation of the component. Fine. Another important factor is the temperature of the column. Temperature is a vital factor in column chromatography because some chromatographic columns are damaged at higher temperatures. Large scale protein purification processes are done at 4 degrees Celsius. Lab scale protein purification is usually done at room temperature. Some chromatographic columns, they get damaged at a higher temperature of around 60 degrees Celsius. So it is advisable to maintain lower temperatures for improving the yield and efficiency of the column chromatography. Fine. Equilibrium and column washing is another factor that affects the column efficiency. Then comes the flow rate. Flow rate can be maintained using a peristaltic pump or a AKTA system. AKTA system is a preparative chromatographic system for laboratory scale protein purification. Fine. So a flow rate can be maintained using any of these two systems. Running a column at a medium flow is recommended because very slow rate can lead to zone spreading and too fast can lead to extensive tailing. Last factor, the last major factor for column efficiency is packing. Proper packing of the column is very important to get good results, and some of the points that we should keep in mind while packing a column are that we need to degas the matrix and the buffers. This will help to remove the air bubbles if trapped any. Fine. Next, we should pour the matrix of the media into the column in a single time. Fine. Third, we should place the column properly. Volume needs to be kept straight. So these were the important factors that affect column efficiency. Let me revise them with you. So the important factors were dimension of the column, temperature of the column, flow rate, packing, particle size of the column, and particle size of the absorbent, and what else? Equilibrium and column washing. So well, that just about covers it. If you have any doubts, please do comment in the comment section. Hope this pandemic gets over soon and we get to meet each other. Till then, stay home, stay safe, and thank you so much for listening to me. Have a good day.